Good morning class, welcome back to microbiology. This is Silvana O'Coney and today's lecture, lecture 2, is about prokaryotic cell structure and function. Prokaryotes differ from eukaryotes in many traits, including size, cell structure, and molecular makeup. And although some cell structures are observed in both prokaryotic and eukaryotic cells, some are unique to the prokaryotes. Prokaryotes are divided into two major groups, bacteria and archaea. And although they're similar in structure, the bacteria and archaea exhibit important differences in their cell walls and in their membranes, although considerably less is known about archaeal cell structure and biochemistry. Now, the bacteria are divided into two broad groups based on their cell wall structure. And these differences in structure correlate with the reaction to what's called the Gram stain procedure. We'll talk more about this later in the lecture, and you'll also learn how to perform the Gram stain reaction in one of our prep classes. You might expect that small, relatively simple organisms like prokaryotes would be fairly uniform in shape and size. But a quick scan of these images of different species of prokaryotes shows that this is not the case, as individual species of prokaryotes offer almost endless variety in terms of morphology. In A, we have an image of Staphylococcus aureus. This bacterium is normally associated with skin and mucous membranes of warm-blooded animals. Staphylococcus aureus is responsible for many human diseases, including wound diseases, pneumonia, abscesses, toxic shock syndrome, urinary tract infections, food poisoning, and surgical infections. It's among the most threatening antibiotic-resistant pathogens known and often referred to as a superbug. In B, we can see the bacterium pictured, and it's called Streptococcus agalactiae. This bacterium is the cause of what are called Group B streptococcal infections. Some people are carriers of this bacterium, which can cause also a variety of diseases, including septicemia, meningitis, and pneumonia. Pregnant women are screened for this bacterium because the newborn infant is particularly susceptible to infection by S. agalactiae, which they would contract from their mother during the birth process. Streptococcus agalactiae can also infect a variety of fish species reared in marine and freshwater aquaculture facilities, and this can result in serious economic losses. In C, we have a common soil bacterium called Bacillus megaterium. In D, Rhodospirillum rubrum, and in E, we can see a bacterium known as Vibrio cholerae. This is the causative agent of the disease called cholera. The differences in shape and size can also be seen here with the actinomyces in A. This group of bacteria more closely resemble fungi than they do bacteria. Mycoplasma pneumoniae in B, which is a group of bacterium that actually lack cell walls, so they're quite unique in that respect. You can see the spiral-shaped spiroplasma in C, and also the very unique square-shaped Walsby's square archaean in E. However, most commonly encountered prokaryotes have one of two shapes. Cocci, which are roughly spherical shaped cells, Note the singular coccus. These can exist as individual cells, but they're also associated with characteristic arrangements that are frequently used in their identification. So, for example, diplococci arise when cocci divide but remain together to form pairs. Long chains of cocci result when cells adhere after repeated divisions in one plane. And this pattern can be seen in the genera Streptococcus, of which Streptococcus agalactiae is an example, but also in the genera Enterococcus and Lactococcus. Some bacteria divide in random planes to generate grape-like clumps of cells. So, for example, Staphylococcus aureus that we saw on the previous slide. Some genera produce 4 cocci in a square, and these are called tetrads, and others produce a cubic configuration of 8 cocci. The other common shape is that of a rod called bacilli, singular bacillus. Bacillus megaterium, the soil bacterium pictured here, is a typical bacterium with a rod shape. Now, the bacilli differ considerably in their length to width ratio. So, for example, the coccobacilli are so short and wide that they resemble cocci. Other shapes are not uncommon. Vibrios, for example, closely resemble the bacilli, but they are comma-shaped. Vibrio cholera, that we saw on the previous slide, causes an infection of the small intestine with severe vomiting and diarrhea, and this leads to severe dehydration. This disease called cholera has killed millions of people around the world during several pandemics, and the bacterium is spread by fecal contamination of drinking water. Then we have spiral-shaped prokaryotes, which can be classified as either spirilla, which are rigid helical bacterial cells with tufts of flagella at one or both ends, or they can be classified as spirochetes. The spirochetes are more flexible and have an internal flagellum. The actinomycetes typically form long filaments called hyphae, and these hyphae might branch to form a network called a mycelium. So in this sense, they're actually similar to the filamentous fungi, which are actually a group of eukaryotes. Then we have other very unusual bacterium, such as the pear-shaped hyphomicrobium, which produces a bud at the end of a long hypha. Another example is Gallionella, which produces non-living stalks. And then we have the flat, square-like archaea that Anthony Walsby found living in salt ponds. Bacteria vary in size as much as they do in shape. Escherichia coli, abbreviated here in the centre of the screen as E. coli, is a rod-shaped bacterium of typical size, about 1.3 to 4 microns in diameter. Near the smaller end of the continuum are members of the genus Mycoplasma, which lack cell walls. And for many years it was thought that they were the smallest bacteria at 0.3 microns, but even smaller nanobacteria have since been discovered that range from 0.2 to 0.05 microns in diameter. The largest photosynthetic bacterium, Oscillatoria, is about 7 microns in diameter, and it's pictured here as being about the same size as a typical red blood cell found in an animal. A huge bacterium has now been identified, which lives in the gut of the brown surgeonfish. It's called Epulopsicum, and it grows as large as 600 by 80 microns. That's nearly as large as a printed hyphen. The viruses, as you can see here, starting with the pox virus, then the influenza virus, the E. coli, bacteriophage, tobacco mosaic virus, and the poliomyelitis virus are generally much, much smaller than the bacteria. The major prokaryotic cell structures are illustrated here, but note that no single bacterial cell possesses all these structures at all times. Some are found only in certain bacterial species, or only under certain conditions, or even in certain phases of the life cycle. But generally speaking, a bacterial cell is always surrounded by an external cell wall, and then just to the inside of the cell wall we have the plasma membrane. The plasma membrane can be, in some species, invaginated to form simple internal membranous structures, for example, such as the light harvesting complex of some photosynthetic bacteria. The genetic material is localized in a discrete region called the nucleoid. Don't confuse that with the nucleus, which is quite different. The nucleoid is not surrounded from the surrounding cytoplasm by membranes. Ribosomes, which we can see, and large masses called inclusion bodies are scattered about the cytoplasm. Many prokaryotes use flagella for locomotion. This particular cell has one flagellum, and many bacteria are surrounded by a capsule or slime layer, which is external to the cell wall. And we'll talk about some of these structures in more detail. So this lecture will describe the major prokaryotic structures in more detail. We'll start with the plasma membrane, then look at internal cell structures, then we'll move out to the cell wall, and finally we'll consider a structure that's unique to bacteria called the endospore. Let's start by looking at prokaryotic cell membranes. Membranes are an absolute requirement for all living organisms. The plasma membrane encompasses the cytoplasm in order for cells to interact in a selective way with their environment, whether that's within the internal environment of an animal, or it may be a less protected and more variable external environment, such as water or soil. Prokaryotes must not only be able to acquire nutrients and eliminate wastes, but they also have to maintain their interior in a constant, highly organised state in the face of external environmental changes. Some prokaryotes also have internal membrane systems, such as those that can use atmospheric nitrogen, that is the nitrifying bacteria, and also the photosynthetic bacteria. So the plasma membrane enables that separation of the cell contents from its environment, but it also acts as a selectively permeable barrier, allowing particular ions and molecules to enter and leave and preventing others. Many substances can't cross the membrane, so the plasma membrane must also aid such movement when necessary, and it does this with the aid of various transport proteins. The plasma membrane is also the location of crucial metabolic processes such as respiration, photosynthesis, synthesis of lipids, and also synthesis of cell wall constituents. The plasma membrane also contains special receptor molecules that help the prokaryotic cell detect and respond to chemicals, and also respond to other organisms in their environment. All membranes from prokaryotic and eukaryotic cells seem to have the same common basic design, but prokaryotic membranes can differ dramatically in terms of the lipids they contain. Indeed, membrane chemistry can actually be used to identify particular species. So let's quickly revise what we know about biological membrane structure. You might remember the fluid mosaic model of membrane structure from your time spent in living systems. You might also remember the lipid bilayer that makes up a biological membrane, and within that lipid bilayer, proteins are embedded. The phospholipids are termed amphipathic, and this is because they have polar hydrophilic heads and non-polar hydrophobic tails, and these tails are insoluble in water. There are two general types of proteins embedded in the membrane, and these types are based on their ability to be separated from the membrane. So the first type are the peripheral membrane proteins. These are loosely connected, they're soluble and easily removed. The second type are the integral membrane proteins. They're tightly connected, they're insoluble in water, and they're not easily extracted. These are also amphipathic molecules, just like phospholipids. The membranes in bacteria differ from eukaryotes in that they lack sterols. Sterols are steroid-containing lipids. For example, the most commonly studied sterol is cholesterol. But even though bacteria lack cholesterol molecules, they do contain sterol-like molecules called hopanoids. And these hopanoids probably stabilise the membrane just like cholesterol does. Hopanoids are of particular interest to geologists who estimate that the total mass of hopanoids in sediments is around 10 to the 12 tonnes. This is about as much as the total mass of organic carbon in all living organisms. And there's also some evidence that's been presented that these hopanoids have contributed significantly to the formation of petroleum. So the emerging picture of bacterial membranes is that of a highly organised asymmetric system, which is also flexible and dynamic. So what about internal membranous structures in prokaryotes? Well, although prokaryotes don't have complex membranous organelles like mitochondria or chloroplasts, internal membrane structures can be observed in some bacteria. So for example, like the parallel membranes which traverse the entire cell of this nitrifying bacterium pictured in A, or even this photosynthetic bacterium with extensive regions of membrane that you can see within the cell, and this provides a larger surface area. Also, infoldings of the plasma membrane are quite common in some bacterial species. One of the distinguishing features of the archaea are their membranes. They differ from both bacteria and eukarya in having branched chain hydrocarbons attached to glycerol by ether links rather than fatty acids such as stearic acid connected by ester links. And as you might expect from their need for stability, the membranes of extreme archaeans such as thermoplasma that grows at over 85 degrees Celsius are almost complete monolayers, such as that pictured to the right side of screen. And you can contrast that to the typical bilayer structure, which you can see to the left of screen.